Hi, my name is William Wildsey. This is the antimicrobial lab that we did for our Bio 111 class by me and Serena Thiessen. We're just going to go over this and get the basics on how this experiment was run and why it's important to understand the concepts of antimicrobials in biological organisms. So, what natural qualities makes things antimicrobial? So, plants and fungi can create chemical compounds called secondary metabolites, and they are added as a defense mechanism that prevent organisms from being attacked and harmed by invading bodies. And then there's the link for that image. And then we have components of fruit extracts and antimicrobial agents. So apples have malic acid, propionic acid, flavonols, flavin 3 ols, and then the phenolic acid and ligands. But bananas, they have potassium and they have a pseudostem that is antimicrobial. They have a gram plus and a gram negative that also affect bacteria's growth. And then grapes have tartic acid, flavanols, flavin threols, much like the apples. And then we have lemons that have a high pH because they're citric and acidic fruits. They're very hospitable to most forms of bacteria because bacteria likes to grow in high pH environments. So low pH acidic tends to destroy them entirely. And then we have a group are tested for antimicrobial properties. We chose apples because due to the oxidization that they appear to have after they've been cut open, we wanted to see if that has any effect, if that's a defense mechanism to prevent um, bacterial growth or like fermentation or anything like that. And then we have lemons. Due to their acidic nature, we pondered if they will be easily to prevent microbial contamination. Like I said, lower pH tends to kill most bacteria from what we looked up. Bananas are known for their high levels of potassium, which could negatively impact microbial growth. It's known that potassium is somewhat radioactive, so even though it's like a small modicum amount, I was just wondered if it was enough to like cause a disruption in the growth of extremely small microorganisms. Grapes have been used in a variety of processes to make alcohol, which usually is very sterile when fermented after it's done. Um, the process is complete, it's usually a very sterile thing. So I know they have the capability to eventually become a very sterile form of liquid. So that was a good one to test. And then we also did oranges, and then oranges are yet another citrus fruit that has always been told, like it's great if you fight against viruses, vitamin C, like that's up for debate in the human body. But we do know that it's very citrus, acidic fruit, and maybe it would be really good at fighting foreign organisms. So the methods on how we went about this experiment was first we started off with a background research. Then we collected samples. We made a hypothesis on whether or not it would be antimicrobial or not. We tested those samples. We gathered and organized our data, and then we did a conclusion and post-experimental scrutiny to try and see what we could do and improve on for next time. So we had step one was we prepped some sterile agar plates and made the starter broth of bacteria that would be used to inoculate every single plate from the DH5 growth from our fridge packets. So we take that vial out. We made sure it was the correct bacteria that we both used in our experiment, and we had to make sure that every single plate was being tested with the exact same form of bacteria to keep the even footing for all of our at-home antimicrobials. So the step two was we had to make the actually snip out the paper filter disc with the hole puncher, and then make the concentration of each fruit, and then place the inoculated agar plates out of the fridge, take them out, thaw them, and then inoculate them with that bacteria. And then a control buffer and antimicrobial filter with kanamycin was also used to be placed along with the samples that had been put with a concentration from the mortar and pestle, so we could have a positive and a negative reaction to compare each of our results to. And then we had to let the plates germinate for about 12 to 8 hours after we got every single one of them set up, 3 to a plate. 3 samples to a plate, and then we had to ensure that they were all marked properly and had a nice, even even temperature and a warm climate on the germination pad to sit, wait, and grow out and see what would grow. And then we observed the filters. If you saw a clear ring like around the kanamycin, we knew that fruit had antimicrobial properties. But if it had been completely enveloped by the bacteria from the DH5 that we put in earlier, we knew that it did not contain anything that would slow down or harm microbes around our plates. So if there was a ring, it would be fine. But if it wasn't, we knew from our control that it is not antimicrobial. So this is an example of what kind of the prep we had to do. We had to make the plates, organize them like that, and then that's the broth that we used to grow out the DH in, so we could inoculate it all. You had to let that grow out first before you could take any other further steps. And then we had to collect our samples, much like this. There's the grape, and there's an orange, and basically what we did was we had this broth, this LB Media broth, and we had our fruit, and you would clean out the mortar and pestle from the test that you just did. You would add the fruit, mush it down into a fine paste in a liquid, and then add a little bit of that, but you wanted to keep a strong concentration of whatever antimicrobial you were testing. So you get like a slurry, kind of like on the left. And then that's what you would dip your filters in, your paper filters in, to get that coated in the fruit. And then you would place that back onto the plates you saw on the earlier slide to let grow with that bacteria. So these are my plates that I had on hand that I did. So I did lemons, apples, and bananas. And as you can see, for all the lemons that I tested marked with an L, that was the only contender that I tested for that actually successfully was antimicrobial. Um, the lemon juice that I got from the plate on there did not allow any bacteria to climb up its way over into and around it. So it's just like the kanamycin where it's basically completely fine. This was after like I believe like 19 hours of letting it sit. Some lemon juice, leaked out from the sides of the paper disc, and that whole area was kind of just non-contaminated with bacteria. The apples, it appeared to be, from my experiment, the most susceptible to the bacteria, getting covered in every single plate that we tested for. And then followed closely after by bananas, which were covered second most out of all of them. But lemons, definitely, every single one had a defensive ring around it. If a large amount of lemon juice was on the plate with it, completely killed all the bacteria around it. Mm-hmm. So this is kind of a graph we have of some of the buffer and controls that we had for the contamination. So we had the 7 live bacteria plates divided in 2 sections that are carefully labeled, antibacterial AB, and then the control 4 out of 7, 57% of the AB control plates remain resilient towards the LB media. And then you have figure B, which is the control, which is going to be always contaminated because it's the control. So there's a little bit of a breakdown for you on what we have for our graphs. All right, and then so our conclusion for this project, we had an antimicrobial lab, came to the conclusion that all our plate samples collected created a ring of antimicrobial defense, where the grapes and lemons were the only 2 fruits that actually had an active defense against them when compared to the buffer and the kanamycin, where they were completely enveloped in the bacteria. So oranges not antimicrobial, grape antimicrobial, lemon antimicrobial, banana not, and an apple were not antimicrobial after all of our testing and data collection. So final slide would be the future improvements. We would like to add more complex concentrations to see if 2 combined antimicrobial fruit if they were synthesized together, would perform better in our testing. We would also like to increase the sample size, of course, to increase the range of fruit that we are testing to ensure better accurate results across more in-depth fruit testing. And then we would also ensure better sterility in further experiments, try to get that down as best as possible so we could have a completely picture perfect plate that had perfect rings around every single sample. And then yeah, we would really enjoy to just keep taking this further and see if we could make like cocktails that could prevent more bacteria from forming than compared to others. That was it for our presentation. It was really fun doing this. If you have any questions, you can leave them under the discussion post. Everybody have a great day.